activity, what we're going to do is we're going to look at binding of a transcription factor data set. So we're asking the question, we have this ChIP-seq data set, and we're asking the question of which promoters, the promoters of which genes are bound by a particular protein. So the one that I'm going to have you guys do a workflow through is NF-kappa B, I believe. And I'm going to show you guys where you can download these data sets and how to read them. And then ultimately we're going to use bed tools to, first of all, create a list of promoter regions from gene information that we have. And then we're going to use bed tools to see which of these promoter regions have binding data for NF-kappa B. And you can use that to give you a list of genes that are bound by NF-kappa B and potentially regulated. So one thing that we've used a lot, but we haven't looked at in detail, are these GTF files, and we're going to take advantage of some features available in here. So I wanted you guys to actually get a chance to look at it and see what it is. We referred to these annotations before. This is the ensemble version. There's a ton of information stored in these files, and you can use it for all sorts of different things. So the way that you read this is each of these sections is associated with the same gene, and so within a gene there could be multiple transcripts depending on the isoforms. So they outline for you the very specific transcripts that are associated with each gene, as well as the coordinates of the exons that go with each transcript. You'll also find UTR information here for genes that have UTRs. So the standard of this, and this is where we're getting into the files in the bed format. I mentioned this on day 1. This is kind of like a useful universal format of files and how you're going to look at regions of genomes. And the standard is always in a tab separated file. You're going to have chromosome, start, stop, plus other stuff. But the chromosome start, stop is where you're going to use to define the regions of where all of these things are located. So you'll see that this gene is located on chromosome 1. It has a start position here and a stop position here. This is the strand that's associated with that gene. And if you continue to scroll over, you can see the gene ID, it tells you the status, it tells you the name, it tells you the type of transcript, it tells you who found, like which database it was curated from. It tells you a ton of stuff, and you can read more about this in the GenCode website to see all of the information that's contained here. So again, the gene has this chromosome start and stop coordinates. The specific transcripts, again, this is alternate isoform information for this gene, have these chromosome start and stop coordinates. And then within each transcript, so this transcript has 3 exons. This is the first exon, it's start and stop. The second exon, it's start and stop. The third exon, it's start and stop. So if you were to look at a cartoon of this gene and say this is, for example, transcript 1, it would look something like this because there's 3 exons in this transcript. The gene start and stop would be here. Because we're looking at this particular isoform, the transcript start and stop would also be here. But then it breaks it down for you on the individual exon level as well, which can be really useful depending on your application. So the first exon start would be here, and the first exon stop would be there. That's the first line in the file. And then the second one defines these coordinates for you, and then the last one defines those coordinates for you. So anytime you hear somebody talking about a bed file is a file defining these regions. It's defining these regions with the chromosome position and the coordinates, and then whatever feature it is that you're looking at. So this example is not in the bed format, it's a GTF format, but it defines the regions of genes and transcripts and exons and all of their other features. The other way that you define regions in bed format is for binding data. So with binding data, when you do these ChIP-seq and whatever seq data experiments, are you guys familiar with seeing these things where they show like this peak of all of these reads that are like upstream of this gene? And this would represent where a particular transcription factor is binding to the genome. Are you guys familiar with seeing stuff like this in papers? People are not as much. Okay, so when people are reporting that their gene is bound to a particular protein or to a particular region, I'll show you we need a way to be able to describe where these proteins are binding. So we have the seq experiments where we can isolate the very specific binding sites, and then we can map that to a reference genome. But ultimately what we care about is where did that read or that section map. So here's my data. So this is an example of, that's a very poor example, of what a typical binding data set looks like when you're looking at the reads and you get these like peaky regions. So remember we talked that this kind of data set is not RNA-seq because we would see big drastic fall-offs around the exons. But instead, when you're doing binding profiles of either RNA or DNA, you get these very peaky regions of where a particular binding site would be. So maybe this is a better example. We see this pretty strong signal right here, meaning a lot of reads are mapping to this very short location in the gene, meaning that I have binding directly at this location. So in order to do some sort of downstream analysis with this, I need to define what this binding location is. So that's what I'm reporting in a bed file. A bed file is going to have multiple lines, and it's got the same format where you have a chromosome start-stop. So I have multiple binding locations across the genome, and they're defined based on their chromosome coordinates. So my first row in my bed file might be, say, chromosome 7, position 10 through position 25. This is one line in my bed file, and it represents one binding site. The next one might be on chromosome 22, position 5 to position 100. These are really small numbers, not what the chromosome coordinates actually would be. But you guys get the idea that each line defines a region of a particular binding site. So we're going to be looking at an NF-kappa B data set, and it's going to be defining the regions that are bound by NF-kappa B. So we know that NF-kappa B would be binding in promoters, so we need to define which promoters it's binding to see which genes it's regulating. So that's the goal of what our bed tools analysis is going to be. Does anybody not understand our goal? So we have data of where NF-kappa B is binding. We know that it's binding in promoters, so we want to look at the promoter regions of genes and see which genes would potentially be regulated by NF-kappa B. Yeah?
How do you regulate the number of copies of gene in a gene?
So that depends. How do you what? What's it?
So normalize. So
Normalize for it?
It appeared to me that some of the peaks where there's binding have more reads than others.
Yeah.
So is there, I guess the question that one, is there a normalizing factor for that? And two, is there a threshold that people have a standard of setting above this amount of reads that would consider that to be where it potentially binds?
Yeah, so that's the way that, and that's more kind of the specific details of a ChIP-seq or a CLIP-seq data set where you're getting these pile up of reads, and that's when people do like peak calling algorithms. There's a whole field on this to identify those regions that have really big high pile-ups of peaks to identify that that's actually a binding site, because you get a ton of noise in these data sets. So identifying which of these regions are actually real binding sets, that's what people in the field do with peak calling algorithms, and that in itself is a whole class. But basically what I need you guys to grasp in order to be able to get this exercise is we do the experiment where we found that. We've found that there's these highly regions that we have lots of reads mapping to, and these are our actual binding sites. And so we're going to define our binding sites in a bed file, and we're going to use that information to determine which genes are bound. Okay. So we're going to use that information, and we're going to use bed tools. So first of all, in order to get the file of transcription factor binding sites, we're going to get it from UCSC. I also included a link down here. This is a good reference point for all of standard file formats. So we talked about BAM, FASTQ, and now BED. But there's a lot of other ones that are less commonly used in the field, and all of the descriptions for how those file formats are and what they mean is located here. Okay, so we're going to go to the table browser on UCSC, and there's a link to it here. And we're going to download a transcription factor chip data set. And this is going to have a lot of different proteins in it. But we're only going to use chromosome 22, so this can be kind of space and memory intensive. And for the purpose of making things go quickly in our class, just define the region as chromosome 22. But if this was something you were doing on your own, obviously you'd use data across the entire genome. So you should be able to follow these instructions very easily in order to get a transcription factor binding data set, and we're going to call it tf.bed. And once you have it, you're going to download it to your computer, but we need it on TSCC. So use SCP to move it to TSCC.